Question 4. Basic agricultural genetics. Start this question on a new page. 4.1. A table below indicates measured pre-weaning growth rates G per day of certain cattle crosses. 4.1.1. Identify the type of breeding system in the table above. The breeding system is called crossbreeding. 4.1.2. Refer to the table above and select the parents that produced calves with the highest average daily gain from pastures in poor condition. Hereford bulls and Brahman cows. 4.1.3 give 2 reasons to support the answer to question 4.1.2. 2 reasons for better performance of these calves are that the offspring have hybrid vigor heterosis. Please note that hybrid vigor is heterosis. Also, the offspring are better adapted to poor veld conditions, more hardy. 4.1.4. Draw a bar graph to indicate the performances of crossings 1, 2, and 9 on poor, average, and good pasture conditions. Okay, sweet stuff. First things first, your graph should have a heading: the performances of crossings 1, 2, and 9 on poor, average, and good pasture conditions. Secondly, it should have an accurate x-axis. Show me that crossings 1, 2, and 9 exist. Also, show your label on the x-axis labeled number of crossings so I know I am dealing with the number of crossings. Thirdly, on the y-axis it should also have accurate things. Show me the correct scale. If you are using multiples of 100, then I should see 0, 100, 200, 300, 400. Or if you're using multiples of 50, then I should see 0, 50, 100, 150, 200, 250, 300, 350, until you reach the maximum number that will cover your whole dataset. After the scale is done, label it on the y-axis and tell me that this scale is for average daily gain, grams per day. Show me on that label that it has units and it is measured in grams per day. Grams per day are the units. Fourth step, then your graph should be a bar graph, really. No funny stories. If they say line graph, you draw a line graph. If they say histogram, you draw a histogram. Okay, they said bar graph, so we gave them one. Remember, because they said for poor, average, and good pasture conditions. This means in the top right corner, you will give us keywords that explain how you differentiate the conditions. Use different sketches in a square box. For poor conditions, use black. For average conditions, use white. And for good conditions, use some lines. Your 3 squares should be different. On the graph, the crossing number 1, make sure you show the average daily gain for poor pasture conditions, represented by black color, and show the average daily gain for average pasture conditions, represented by white color. Also show the average daily gain for good pasture conditions, represented by line crossings. Do the same thing for crossing 2 and 9. Now that we have done the graph, check the accuracy. If it is accurate and all other steps were done very well, then you got 6 marks. 4.2. 2 pairs of genes control the milk production of a Jersey cow. The genotype AABB yields 200 liters of milk. Each additive dominant gene adds 20 liters of milk. 4.2.1. Indicate the type of inheritance that controls the milk yield of a Jersey cow. The type of inheritance that controls the milk yield is polygenic inheritance. 4.2.2. Calculate the milk yield of a Jersey cow with the AABB genotype. The milk yield of a Jersey cow with genotype AABB is 2 dominant AA, which gives us 20 plus 20. The sum of 20 liters and 20 liters is 40 liters. We know that the genotype AABB yields 200 liters of milk. Because now we have 2 dominant alleles, giving us an additional 40 liters. We will say 200 liters, which is the base yield, plus the additional 40 liters. After calculating, it gives us 240 liters as the final answer. 4.2.3. Work out the genotypic and phenotypic ratios of the F1 generation if the cross between AABB and AABB is done. Phenotypic and genotypic ratios of the F1 generation homozygous dominant AABB is crossed with a homozygous recessive AABB. We are going to have our gametes from each parent. On the left side, we have the dominant allele AB, and on the right, the recessive allele AB. After crossing them, we have 4 offspring with the heterozygous alleles. The phenotype is that all our offspring produce 240 liters of milk. 4.3. The picture below indicates a pattern of inheritance in farm animals. 4.3.1. Identify the phenomenon above. The phenomenon in question 4.3 is called atavism. 4.3.2. Give a reason for the answer to question 4.3.1. The reason is that a recessive gene for red, which was switched off and not expressed in the phenotype in the past, is now switched on and expressed. 4.3.3. Give the alternative name of the phenomenon in question 4.3.1. The alternative term for atavism is throwback. 4.4. Three important aspects are considered when selecting individuals for breeding programs, namely biometrics, heritability, and estimated breeding values. Selected individuals may be mated using various breeding systems, such as outcrossing, species crossing, and crossbreeding. 4.4.1. Differentiate between selection and heritability. Selection is the process of choosing individuals for breeding based on superior characteristics. Heritability is the degree to which genetic factors determine the characteristics. 4.4.2. State two advantages of species crossing. Results in hardy animals and drought animals. 4.4.3. State two related breeding systems not mentioned in the case study. Line breeding and inbreeding are two related breeding systems. 4.4.4. Explain the importance of using estimated breeding values in breeding. Using EBV is important because it indicates the heritability of a particular characteristic to predict the success of a breeding program. 4.5. Mutagens change the genetic material of an organism, causing errors in the genes of organisms. How will the following mutagenic agents affect the DNA structure of the gene? 4.5.1. Gamma and X-rays. Gamma and X-rays damage DNA, causing it to break. 4.5.2. Metals such as nickel and chromium. Metals change the chemical structure of a DNA molecule. 4.5.3. Alkaloids. Alkaloids prevent chromosome segregation. 4.5.4. Viruses. Viruses insert their own DNA,